Thank you very much, Natalia. I think I elevate these a little bit. I really appreciate being invited by Ann and I to share this with my distinguished co-chairs, Alan Walker and Ray Ogra. It's been a pleasure to work with them to get this program together. You heard about one single protein this morning. Nick Embleton talked about lactoferrin. I will talk about osteopontin, which I think is a little bit more new on the radar screen for most of you. And I will try to tell you a bit about osteopontin and what it possibly can do. Osteopontin is a very acidic phosphorylated glycoprotein and extensively post-translationally modified. There's about 28-30 different phosphate groups, and there are several glycans along the molecule. And more importantly, osteopontin contains an arginine glycine aspartate, or what is abbreviated for the amino acids RGD domain, which is actually an integrin binding site that works as a receptor for osteopontin, and also a heparin binding domain, which is recognized by the CD44 receptor. So osteopontin has several receptors on the cell surface. It's synthesized by a variety of tissues, including the mammary gland, brain, bone, immune organs, and intestine. And as you can recognize, it was first localized in bone. That's because that's how it got its name, of course. It's present in most body fluids, such as milk, serum, and CSF. It's involved in a lot of biological functions: cell proliferation, cell survival, biomineralization, again coming back to bone, but many of them in immune function: leukocyte recruitment, inflammation, immune modulatory functions, wound healing, and initiation of cell-mediated immune responses. And I will show you some examples of this later on. We started analyzing osteopontin in breast milk. This is a collection of an NIH-sponsored breast milk study that I did some time ago, and we used that milk bank to look at osteopontin concentrations. And as you can see, it's very high the first couple of weeks, colostrum, but also in transitional milk, and then it maintains a stable but relatively high level up to 1 year of lactation. And it's actually the 5th or 6th most abundant human whey protein. So it's kind of surprising that you see so little of it in the literature. And it's very high. Like I said, this came out a few years ago from a Danish group of researchers showing that colostrum, again, human colostrum, is very high in osteopontin. Term milk is lower. But in particular, cow's milk is low in osteopontin. And not only that, but infant formula is even lower. During the processing, where you take skim milk and whey proteins and make it into infant formula, osteopontin is partially lost. So it's a very low concentration of osteopontin. This Danish group, they exposed osteopontin to gastric juice and found out that it seemed like it was resisting proteolysis. And later on, it was found that milk osteopontin can survive in vivo proteolysis in 20 days old mouse pups. And we have verified that, as you will see later on. A few years ago, bovine osteopontin became commercially available from Arla Foods in Denmark, and therefore the possibility existed to add it to infant formula. And why would you do that? Well, like I said, the amino acid composition of the bovine form of osteopontin in human is partially different. There's about 70% homology, but the phosphorylation regions are very similar. The glycosylation regions are similar, even if the individual monosaccharides vary a bit. But importantly, the integrin binding sites and the polyaspartic acid regions are totally conserved between human and bovine osteopontin. So both of them can bind to the same types of cellular receptors. We did a preliminary trial on this bovine osteopontin. This was done together with Sharon Donovan, one of my previous PhD students. And we fed infant formula with bovine osteopontin to infant rhesus monkeys. We believe that this is a very, very nice model for the human infant. Rhesus monkey milk composition is very similar to that of human milk. We can use it. They can get formula and feed themselves from basically when they're born, and they are weaned at about the same age as human infants, about 4 to 6 months. So this was a limited number of monkeys. As you can see, there were 4 breastfed ones, and then we had 6 each given formula without osteopontin and with osteopontin at the same concentration, roughly, as you have in breast milk. And in this case, which we unfortunately, of course, not can do in our studies on term infants, we got the jejunal messenger RNA, so we could do transcriptomics on the intestinal samples. There were no adverse effects at all. Growth and clinical parameters were similar. Breastfed monkeys were a little bit leaner, which they often are. And what we showed in the gene arrays was that there were 1,017 genes that were different between the breastfed and the formula-fed infants. But when bovine osteopontin was added to the formula, this diminished to 200. So it made them remarkably more similar to breastfed infants. And then there were, of course, some differences between the regular formula ones and those that were fed formula with bovine osteopontin. And we concluded from this one that it's safe, and of course we were not too concerned about taking a regular dairy fraction and adding it to cow's milk formula. But we believed that the preliminary data from the infant monkey said that we could potentially have biological activities by adding this osteopontin. Because we saw these differences in gene expression profiles. The pathway analysis I don't show here, but the pathways mostly affected by the bovine osteopontin was those related to immunomodulatory functions and to cell proliferation and differentiation. I then had the opportunity to do a clinical study where we added, similar to what we had done in the infant rhesus monkeys, we added bovine osteopontin to infant formula. This was done at Fudan University at Shanghai in China. Dr. Peng did a fantastic job on this study. It's a good site to do clinical studies, similar to what Ola and I have done in Umeå. Virtually all the infants there in Shanghai, in that area of the city, are breastfed from birth. About 80% are breastfed at 1 month of age. So that means that it's harder to find the formula-fed infants than it is to find the breastfed infants. But they have 10,000 births at that hospital, and therefore recruitment rate was quite decent. The formula was done by a Chinese company called Biostime, and Arla was providing the bovine osteopontin. In this case, we were fortunate because we were allowed to use 2 concentrations of osteopontin. As you can see, in very many of these clinical trials, only 1 dose level is chosen because these are work-intensive, they're expensive, and a lot of times you can just have 1 concentration. Osteopontin this far is relatively expensive to add to formula, and therefore it would be of interest to see if a lower level than what you see on an average in breast milk would have similar effects or not. So this is the breast milk average level. This is about half that level. But with the individual variation among women, there are women that will have this level in breast milk also. We had a regular infant formula without bovine osteopontin and with a reference group of breastfed infants. And this was a randomized controlled trial, double-blind. And we did, of course, anthropometry. We measured it at birth and every month up to 6 months of age. We took venous blood at 1, 4, and 6 months of age, and the parents, together actually with the clinical staff, they visited them to make sure that the information was obtained properly. There was a feeding and dietary journal where they recorded formula intake, and also there was a very detailed morbidity journal that was supervised by the clinician also. We got tolerance data, stool characteristics, etc. We analyzed hematology, and we did flow cytometry at Fudan University. We got red blood cells that we sent to Sharon at Illinois, so we have done transcriptomics on those. That has not been published yet. We did serum ferritin at UCD, plasma amino acids, we did BUN, and we did an extensive battery of cytokines, also at UCD. And this one is a busy one. The bottom line here is I just want to show you that we had very low dropout rates. Ole showed some studies that have been done by other investigators where you have dropout rates of about 40–50%, and you'll start wondering how valid the data will be. We had less than 10% dropouts average after the 6-month observation period, which I think speaks really well for both the clinical investigator and her staff at Fudan University. Parents had 24/7 access to the physicians and nurses by telephone and home visits. We didn't see any significant differences in weight gain, nor did we see any difference in height. We also didn't have any significant difference in adverse effects. In fact, they were very low. There were no serious adverse effects, and the number of adverse effects was low and not significantly different. No difference in appetite and formula volume consumed, nor in sleeping time. I'm skipping a lot of the other clinical outcomes, but I would like to emphasize something here. And something that a lot of clinicians and nutritionists alike are not aware of is that infants fed formula have significantly higher TNF-alpha. In this case, it was not significant, but Erika Isolauri, who is at the same institute as Sameli Rautava in Turku, they have shown that formula-fed infants have higher circulating levels of TNF-alpha. And to me, that signifies that it's something we're not doing quite right with these. I think it's a signal that something may be going on down in the gut, which is a little bit out of whack. And if we could do something to normalize these levels, I think it is most likely going to be beneficial. Here you can see the TNF-alpha. This is confirming what you've seen in Turku. And you can see here that even if the baseline studies were taken at 1 month, when the mothers stopped breastfeeding and started with formula, they started with the respective formulas that they were assigned to by the randomized controlled design. And here you can see at 4 months, and this was exclusive formula feeding and exclusive breastfeeding up to 4 months. After that, and this was the intervention, after that they actually got some weaning foods. But you can see that with the repeated measures analysis, those infants that received bovine osteopontin at the higher dose here, they were significantly lower than those fed regular formula, and they were not significantly different from the breastfed ones. By 6 months, it was different, but like I said, at this time they had stopped having the formula, and they were getting weaning foods, and there were no significant differences whatsoever. But it seems like it may be a dose response. We didn't see a significant difference here, but we saw it for the higher dose. We did several inflammatory cytokines, and the picture is similar. I will not show those. We also looked at anti-inflammatory cytokines, and the same thing happened there. The profile of cytokines became more similar to breastfed infants than those that were fed formula without osteopontin. Well, how does this translate into morbidity? Well, we registered days with fever, and here you can see, similar to what Olle showed for Sweden, it's a higher incidence of fever in those fed regular formula than in breastfed infants, and then adding bovine osteopontin at either of these two levels made them not significant from breastfed infants. So a reduction in days with fever. So it may be that the immune competence was altered to make them more similar to breastfed infants and more perhaps capable of fighting infections. We had subgroups of different types of infections. We did not have otitis media registered in this study, and we didn't see any individual group differences within each category. We also did very extensive, like I said, we did flow cytometry. Tina West, who is also from University of Umeå in Sweden, was in charge of that. And we looked at a lot of different immune classes. And what we found, to make this short, was that the group that got the high level of osteopontin had an increased proportion of T cells as compared to a standard formula, and those that were fed half that level of osteopontin. We also had effects on the monocytes. There were other subgroups that were altered also. But we just concluded, and this was published 2 years ago, that osteopontin in infant formula at the higher dose level made the infants similar when it came to the distribution of different subclasses of immune cells, and therefore may affect immune function. So to conclude from this clinical trial, and this to date is the only one that has been done in infants, more studies certainly need to be done. Addition of osteopontin resulted in no significant differences in adverse effects, formula intake, growth, or sleep, as compared to those fed regular formula. Immune cell distribution and cytokine responses were more similar between infants fed the higher level of osteopontin, the breast milk level, and the breastfed infants. Days with fever was higher in infants fed regular formula, but not in those that were given bovine osteopontin. And therefore, we concluded that feeding infants formula with added bovine osteopontin resulted in outcomes more similar to those of breastfed infants. With that, we moved on, and we actually analyzed osteopontin in the plasma of these infants. And here you can see that already at 1 month, there were apparent differences when it comes to human osteopontin in the 4 different groups. These are the breastfed infants, these are the infants fed bovine osteopontin at the 2 levels, and those were the infants fed regular formula. This became highly significant at 4 months of age, where they have been exclusively fed breast milk or regular formula or formula with bovine osteopontin. And this persisted up to 2 months after the intervention. And this made us to hypothesize, and you will see from the mouse that it's there's most likely autocrine upregulation of endogenous synthesis of human osteopontin caused by bovine osteopontin. And here you can see the analysis of bovine osteopontin in the serum of these, or the plasma of these infants. And you can see that at each sampling occasion, there was much higher levels of bovine osteopontin in the plasma when the formula contained bovine osteopontin. Note the scale. These levels of bovine osteopontin are considerably lower than those of human osteopontin, but it shows that certainly there was sampling of the bovine osteopontin, and we know that bovine osteopontin enters the enterocyte. We've done confocal microscopy. So some of the bovine osteopontin goes into the systemic circulation. Then we wanted to explore in the laboratory what the biological functions of milk osteopontin are, and we used a knockout mouse model, which I will explain in a few seconds. The reason we did that was that if you go in and do a PubMed on osteopontin, you will get a couple of thousand responses. It's in bone metabolism, it's in cancer, it's in just about anything. But if you go in and search for milk osteopontin, I think there is by now 5 publications. So we really don't know what the osteopontin presented to a newborn does to the newborn. We hypothesized from the literature that osteopontin may not only have effects on the immune system, but also on the brain and the development of the gut. And what we used here is, like I said, a knockout mouse model. And it's important here to recognize what we really did. We had wild-type mouse dams with wild-type pups. And then we had knockout dams that produces milk, which is totally void of osteopontin. And she fed normal wild-type mice. So they have the capacity to endogenously synthesize osteopontin in all tissues, just like normal, but they don't get that source of milk osteopontin. And then we did a whole battery of tests. What I will show you first is it's important when you have a knockout model that we didn't have an effect on milk yield, that they are lactating normally. We looked at the organ distribution in the pups by radiolabeling. And then we had these 2 groups that I just mentioned, those that were osteopontin negative, that didn't get osteopontin in the milk, and those that got osteopontin in the milk. We looked at intestinal morphology. We gave them an LPS challenge, and we looked at cytokine response to the challenge. We looked at normal immune function by cytokines, and we looked at the brains, and we also did a battery of cognitive tests to evaluate their brain function. And the first part of this was the brain. What does milk osteopontin do on the brain? It is abundantly expressed in the brain, and in rat brains it's been shown that osteopontin-expressing cells appear from embryonic day 16 and increase remarkably during the first week of postnatal life. And osteopontin has been shown to stimulate proliferation of neural progenitor cells isolated from adult rat brain. Again, very little have they looked at the newborn animal. And it's a well-known regulator of myelination, which of course is important for brain function and cognition and cognitive development. Here you can see, like I said, we showed that milk yield was identical in the wild type and the knockout dams. We confirmed, we killed sequentially some pups that were fed by wild-type dams, so they got osteopontin in the milk. And here is Western blotting using antibody against mouse osteopontin. And it's a little bit reverse here. You have the mouse way here with osteopontin at a couple of different molecular sizes. And here you can see in the stomach, it remains. Pepsin doesn't do a whole lot. And then you see some fragmentation to larger osteopontin fragments in the duodenum, the jejunum, and the ileum, so the first, second, and third part of the small intestine. So it does really survive throughout the small intestine in relatively large fragments, and these fragments do contain the receptor binding sites. We didn't see any significant effect on, weight gain or body weight or brain weights in those that got milk osteopontin and not. We did histology. We didn't see any apparent differences in morphology between the animals. We looked at the expression of osteopontin in the brain, nothing significant. But when we did Western blotting, of osteopontin in the brain, here you can see that we had highly significant differences. It's a little bit busy slide, but you have days 1, 2, 4 here, 6, 8, 10 here, and then you have those that got osteopontin in the milk, those that didn't get here matched. And you can see that there's a dramatic difference, particularly between days 6, 8, and perhaps also 10, between those that have been fed milk osteopontin and not. And then we went on, like I said, and did testing. And for those of you that are not familiar with this, this is the passive avoidance test, very popular to use. It tests more memory function than anything else. And you have a device here where the mouse pup will be put into the white compartment. And the natural tendency for the mouse pup there is to go into the dark compartment. They want to hide. Unfortunately, once they get in there, they get a small electrical shock, which they don't like, and they go out. And the natural tendency then for the normal mouse pup is that if you do that 1 or 2 times, you learn and you don't go in there to get shocked again. But those pups that didn't get osteopontin in the milk had a significantly poorer performance. They just couldn't remember as well that they got an electric shock there. So they tried again and they tried again. And this is showing that one part of cognitive function was impaired if you do not get milk osteopontin. We did another one which is a little bit more related to both memory, learning, and physical capacity. It's a rotarod test, and it's a rotating apparatus where the mouse is put on top of these rotating wheels. And again, if they learn, they are quite capable of kind of balancing that. They're rotating very slowly and successively going faster, and then the mice learn how to cope with that. But if you don't get milk osteopontin, you don't learn as well. Again, showing that cognitive development and memory functions seem to be impaired if you don't get osteopontin in your milk. Myelination, of course, is very important. You form the myelin sheath around the nerve to allow nerve impulses to move quickly. And it's a very complex process, and I'm not an expert on this at all, but it's really important for the development of proper function of particularly oligodendrocytes. And osteopontin is normally secreted by astrocytes and microglial cells and may stimulate proliferation of these precursor cells to promote myelination. And this is just a graphic picture showing some of these proteins that are involved in this. And you will see they are abbreviated here, but you have spelled it out in the next slide with the gene expression studies in the brain. And here you can see that several of the proteins that are involved here, cyclic nucleotide 3-phosphodiesterase, myelin-associated glycoprotein, myelin oligodendrocyte glycoprotein, and in particular what many use to check this myelin basic protein. And you can see that these are normalized, so 1.0 here represent if you have normal in both the osteopontin plus and osteopontin minus mice. But here you can see that in the brain of the osteopontin negative ones that didn't get osteopontin, that the gene expression of virtually all of these is significantly reduced around day 6, 8, 10 as compared to normal. So very specific molecular events taking place in the machinery involved in myelination, which may explain why we saw these functional deficits in the mouse pups. So milk osteopontin increases osteopontin expression in the brain. That's why I said it's most likely some type of autocrine regulation there. And the upregulated osteopontin expression may increase myelination and promote brain development. In retrospect, of course, it would have been very nice to have done Bailey test in the study we did in China, but at that time there was no suggestion even that milk osteopontin may have an effect on the newborn brain development. And the next study definitely should, in my opinion, have that included. Then we looked at immune development. And osteopontin is involved in the initiation of cell-mediated immune responses and promotes a Th1 cytokine picture and inhibits Th2 cytokines. It plays an important role in both acute and chronic inflammation, and it may exert both pro-inflammatory and anti-inflammatory actions depending on the nature of the biological scenario. This was from a review article from adult experimental animals. So what we did to mimic an infection, we gave LPS, which you have heard about earlier, so I won't go into that. And it's known that LPS can upregulate OPN expression in macrophages through TLR4 that we heard about yesterday. Not about osteopontin, but TLR4. And here you can see the results in the LPS-infected animals. And you can see here that interferon gamma, 4 hours after the LPS, was significantly lower in those that had not been fed osteopontin in their milk. And this was accentuated at 8 hours. And you can see the difference. We tested them at day 20, day 30, and day 40. And it was particularly pronounced here in the day 30 animals at 4 hours, and at 8 hours in the day 20 and day 40 animals. TNF alpha was the opposite, and there you had a significantly higher TNF alpha response in those that had been fed osteopontin deficient milk. So a downregulation of TNF alpha when osteopontin was in the milk, suggesting that pro-inflammatory cytokines may be downregulated by the milk osteopontin. So therefore, milk osteopontin may contribute to resistance to LPS administration and therefore bacterial infection by altering immune responses. More work needs to be done on that, and we're doing that as we speak. So, to conclude, I would like to say that milk osteopontin plays important roles in brain, immunological, and gut development during early life. Breast milk osteopontin likely provides beneficial bioactivities to the breastfed infant. And I think that is what we learned from the clinical trial. It suggests that if you would add something like bovine osteopontin that has similar binding sites as the breast milk osteopontin, you may be able to provide some of these bioactivities. And I think we need to explore much more what the osteopontin in the breast milk is doing to the breastfed infant, but that is more difficult to assess. Okay, with that, I would like to thank my collaborators, Dr. Jiang and Dr. Christine Prell, who did a postdoc with me from München. And with that, I'll end my presentation. Questions? Yes, Dr. Ogra.
Bo, that was a wonderful presentation, really. Outstanding. Thank you. Because we don't know much about osteopontin at all. My question relates to the effects on the brain itself. Does the osteopontin which you give by mouth get into the central nervous system, or it stays in the gut and it interacts with the gut enteric nervous system?
No, we have done radio labeling and we can detect it in the brain, yes.
How does it get there?
That's a good question. We see it in serum, definitely. If it is then, and others have reported that it passes the gut, the blood-brain barrier.
I see.
If there are other routes to reach the brain, I don't know.
Well, I was wondering about the gut-enteric nervous system, whether you produce a lot of, whether the osteopontin induces any neuropeptides in the gut, which may be stimulating the enteric nervous system via different pathways.
I wouldn't exclude that possibility for sure.
Have you looked at any neuropeptide synthesis in the gut?
No.
Serotonin or anything else? No?
We will do that, but we haven't done it. Thank you.
Okay.
Yes, Vanessa.
So on your bovine osteopontin trial, you showed one slide. Did it indicate there was endogenous osteopontin or induction of endogenous osteopontin production? Right. So have you thought about teasing out the effects of that osteopontin intervention as being directly related to the bovine osteopontin provided, or more directly related to the induction of endogenous production? So if you're looking at osteopontin-specific responses, do you think it has more to do with that the increased endogenous production in those mice receiving the bovine osteopontin? Does that make sense?
No.
It looked like there was a difference in the endogenous production in the mice that were provided the bovine.
Yes, that's correct.
So
when they got milk osteopontin, they got increased synthesis of brain osteopontin. Yes.
Right.
Yeah.
So
and in the human, we saw higher levels of circulating human osteopontin when they got bovine osteopontin.
I guess that's kind of my question, is perhaps the provision of the bovine osteopontin is maybe the effects you're seeing are more due to the stimulation of endogenous production of that osteopontin, and then the body's response to that, instead of the provided bovine protein itself.
In the human, you mean?
Yeah.
Yeah, that's possible. And that's why I said it's harder to tease it out in the human, where we have limited possibilities of chasing it. Yes, Dr. Oftedal.
Do you know how widely distributed osteopontin is across mammals? I mean,
no.
Don't know.
No, I don't know. I only know that it's found certainly in cow's milk. In mouse milk, it's similar to what we've seen for lactoferrin. Mouse is a high lactoferrin species, and it's also high osteopontin one. And the relative proportion of osteopontin in mouse milk is similar to human milk. We have, as of 2 weeks ago, we have found osteopontin in goat's milk, but we haven't looked at any other species.
And you said you did the monkey trial, didn't you?
We did, but we have not analyzed.
Have not looked at it.
No.
Okay.
No. But it's highly likely that so far all the milk proteins that are in rhesus monkey milk do cross-react with the human antibodies, and I would guess that the rhesus osteopontin, if it's there, which I think it is, would cross-react with the human one, so we could check that, yes.
Okay, and do you know whether it's found in other secretions?
Others have reported in other types of fluids, yes, but we have not looked at them.
Okay, thank you.
Thank you.
Did you look for or have any measurable amount of bovine osteopontin residual or innate into the regular formula as the control arm?
I didn't understand that.
The regular formula that was your control arm?
Yeah.
Was there any measurable innate osteopontin?
Yes, it was the concentration in that one was about 10 milligrams per liter as compared to the 65 and 130. Yeah.
Thank you.
Okay, thank you so much.